So I'd like to, of course, thank everybody, the organizers, and Tomas for giving me this opportunity. Because I was just talking to Jack, and Jack is skeptical of philosophy. And I was a philosophy minor as an undergraduate. So this is a really fun thing for me. I've really had, except in Sorbonne, never had an opportunity to talk about philosophy. So what I'd like to talk to you about today are what we're learning as biologists, and how, as biologists, we are really going to have to turn the way we think upside down because of new knowledge. And so it's the consequences of living in a microbial world and what that means. So what I'm going to do is I'm going to talk about this huge sea change in the field of biology. So there was, of course, a big revolution in the 19th century with Charles Darwin and the theory of evolution. There was one in the 20th century with Watson and Crick finding DNA to be the basis for genetics. And then this is the 21st century revolution, is the understanding of the microbial world. And so it's about getting a better and more realistic grasp of life's diversity. So I'm going to talk about what the basis of the revolution is, like how did we get here, and why it is critical. So first context, and then because Tomas is here, and he's done so much fabulous work on thinking about the immune system, I'd like to give an example of the conspicuous impact it's had on some of the concepts of immunology. So I like to make this analogy because I think it's very apt. And that is revolutions can often come with breakthroughs in technology. And so in this case that I'm showing here, Copernicus had this idea. He had this really interesting idea. It's not the Earth that's at the center of our solar system, but actually the sun. But he didn't really have any great proof of it until about 60 years later, when Galileo's telescope provided the evidence for that. And so we went from being geocentric to being heliocentric. And of course, Galileo wasn't he thrown in jail for this? For something, the Pope did something. Anyway, so, but this invention changed our view of the universe and our view of our position in the universe. So this was a huge shift in the way astronomy worked. What about biology? For over 2,000 years, biologists classified the world based on optics, what you could see either with the unaided eye or with the aided eye. So going back to Aristotle, it was animals and plants. And then Anton van Leeuwenhoek took a scraping of the inside of his cheek and put it on the little microscope that he invented, and looked at it and saw things that he called animalcules, which were microbes actually. So, but he called them animals, plants, and animalcules. Then another step in having the aided eye was another big transition was with the electron microscope. And Robert Whittaker, back around the 1970s, 60s and 70s, built something or developed something called the 5 kingdom model. And that was animals, plants, fungi, protists, which were organisms that had complex cells with a nucleus and other organelles. And then there were bacteria that had no organelles inside of them. How many of you have had the 5 kingdom model? Yeah, when you took biology. It's really interesting because to this day, even when I'm talking to young groups, like incoming graduate students and things, I ask that question. And it's always 50% of the people. So I guess my point there is our message we need to get through a little bit better. Because what has happened is we have a new lens. And this was introduced to us by a guy named Carl Woese back at the University of Illinois. Carl Woese is actually had the idea in the 1970s that instead of using optics, we should use genetic sequences, which are a true reflection of the relatedness of organisms. So Carl Woese began to look at sequences and find out that things were not organized at all like we thought. In other words, optics, it turns out that optics is great for animals and plants. So when I was a kid using the 5 kingdom model, animals, there were 30 to 40 animal phyla that were put into those phyla based on the optics. And when you impose the genetics on those 30 to 40 animal phyla, you got the same answer. And it was because the way they look is very much reflective of their genetics. The problem with microbes is they're, excuse me to the microbiologists in the room, they don't have a lot of features that would allow you to bend them into different areas, or round or spiral or comma shaped or whatever. So, but Carl Woese began to sequence a whole bunch of different microbes, and he found some really, really interesting things. And so the problem was, though, that in his time, up until around 2004 to 2008, when next generation sequencing came in, it was very, very slow going and very expensive. So not a lot of people could do it. You know, it was just something that Carl Woese, and he came out with a paper in 1990, in which he binned organisms into bacteria, archaea, and eukarya, which were, eukarya was the animals, plants, fungi, and the complex unicellular organisms. But we've even come farther than that at this point. And the reason is because after his 1990 paper, the cost of sequencing of nucleic acids kept going down and down and down, and sort of adhering to Moore's law for a long time. And then all of a sudden, next generation sequencing came in, and it went down exponentially. It just really fell. So it went from $6,000 a megabase to something like 3.5 cents. And Rob, what is it now? A megabase?
I don't remember.
Yeah, but it's less than 3.5 cents, I think, because that was like in,
Yeah, it's about $2,500 for 25 billion sequences, 2 by 150.
Wow.
So you can figure it out.
So the point is that it became this. So this was incredibly democratizing. So everybody and their mother in the field of biology went out and began to sequence everything. And so this is what has happened is because of this, we have a whole new view. And so Carl Woese was the nucleating site to this. And then next-gen sequencing was invented. And this invention has changed our view of the biosphere, just like it changed our view of our solar system. This has changed our view of the biosphere, and it has changed our view of our position in the biosphere. So what do I mean by that? What have we learned thus far? First of all, this is that 5 kingdom model. Is there a pointer? No. This is that 5 kingdom model. And so you can see that up there, so the animals, plants, fungi, and protists. And the bacteria are down at the bottom. It was thought in 1970 that there were 7,000 species of bacteria, which is very few. In fact, it's the same number of species as there are snakes. And so there aren't very many. It wasn't thought that there were very many bacteria. And so it was thought that while they had been around for 3.7 to 4 billion years, the microbe and the microbe dominance of the biosphere had been supplanted by animals, plants, and fungi. And that was the idea. But what we found since sequencing is the following. So what we now know is that the eukarya, from what most people are believing at this point, are deeply embedded within one of 2 domains of this group here. So this group is now everything, except these guys are embedded in it. And we now know we've been able to sequence huge numbers of bacteria. As you guys probably know, it's very hard to assign species to bacteria because they trade DNA around and there are strains and you have to do special things to figure out whether or not it's a species. Rob, what is the number of species at this point of bacteria in archaea? Do you have a?
We don't track that.
Yeah, yeah, and part of the reason why you don't track it is probably because it's going so fast and it's just so many areas haven't been explored yet.
Well, it's also not a very useful concept pragmatically for identifying biomarkers or for like there's also a lot of controversy about what makes a microbial species.
Yeah, that's what. Yeah, that's what I meant by saying, we just really don't do that very well. But certainly they're by far and away the greatest diversity of the biosphere. There's no question about that now. So it's overwhelmingly microbial, our world is. And so what we found in all this sequencing and all this democratizing, everything that's gone on, that the activities of microbes drive the form and function of the biosphere, and they're pivotal to shaping all health. Everything from human health, as we all know now, to the health of the soil, to the health of the oceans, and so on and so forth, are in the bacteria in the hands of microbes. And the impact of their diversity and ecological implications of this diversity will continue to reshape. I mean, it's a hard lesson because we're very embedded in the 5 kingdom model in our heads. And so this is something that we really have to change in the way we think. So one of the things I love to do is to say, how much has gone on in this area since 2006? In other words, when next-gen sequencing became really a popular thing. And so what I do is I go to the NIH search engine, which is called PubMed, and I type in the word microbiome and a word. And in this case, I did 4 very different things: the soil, plants, ecology, and the brain. And how many publications have come out in this, and how recent are they? And this is what these graphs look like. So 2008 is where that little red arrow is. And so for the soil, there's a total of 23,000. And in the last 3.5 years, 64% of those have been published. So you can see how enabling this has been to the community, and similarly for plants, ecology, and the brain. It's really interesting. I had the great privilege of being asked if I would consider being the Dean of Biology at UC San Diego. And I was down there in 2011. And there was a very famous group of neurobiologists on the search committee. And I told them what I wanted to do for biology there. And I thought UC San Diego was the best place in the world to do that. And that was to integrate macro and microbiology. And that way we would, people wouldn't get a biology degree and come out with a single lecture on the microbial world at the end of their degree, because we now know microbes are so important. But there was a guy, very, very famous guy from Salk Institute, a neurobiologist, who looked over at me and said, the microbes have nothing to do with the brain. And I looked at him and I said, I'm teaching a course. I was a Moore professor up at Caltech at the time, and I was teaching a course with a guy up there, Sarkis Mazmanian. And we had just covered the very first paper with this class. We had covered the very first paper on the impact of the microbiome on the brain of mice in development, and showing that paper showed that normal development required the presence of the microbiome. And so I said to this guy, I said, you might keep an open mind. I think you might want to rethink that in a few years. And what we've seen is that not only seems to shape the brain, but also there are seem to be a lot of neurological diseases and various things that can go on with the brain that have a component of the microbiome. Okay. The other thing that we have found is that symbiosis is very prevalent. It seems to run the biosphere. And one of the things that I've done, one of the other things I do is I look at textbooks. So one of the most recent introductory biology textbooks, I went in and I had a look at this textbook. And in this textbook is 1,254 pages long. And it's a very famous textbook, very widely used. And there are 6 pages in that book on symbiosis. And it has now become a really big thing because it's, animals and plants are run with, in our case, it's probably, vertebrates have 10 organ systems. It's like the 11th organ system. It's in the definition of an organ system is a group of cells that work together, and the gut microbiome definitely works with the lung microbiome and skin microbiome. But what I'm showing here is obviously I'm showing coral reefs, which are run by zooxanthellae. In the upper right, that's one of the dry valleys of the Antarctic. The only living systems in the dry valleys of the Antarctic are lichens. And lichens, as you guys probably know, are symbiotic systems among algae and fungi and bacteria. There are lots of, there's a lot of work on those. What I'm showing in the lower left there is a montane forest. And the soil, the mycorrhizae, the fungi in the soil were absolutely essential for the water to land transition in plants. Something like 80 to 90% of the plants have fungi, and those that don't have it have lost it. And so those fungi control the water in the soil. They control the delivery of plants. They enable plants to live on land by mobilizing phosphorus from the soil. And so there's another example of, it being really prevalent and important. And then finally, I show this cute little boy and his cat, and both of them have really, really big microbiomes that are critical for their health. So these, we need to begin to integrate these ideas into the way we think about how these organisms work. So, like I said, it's not yet recognized in bio textbooks, which makes me crazy. So Rob is going to be talking to you, I'm sure, about the human microbiome. And we know that the human microbiome is incredibly complex. But we had a 10-year human microbiome project through the NIH, which characterized the human microbiome. So it's probably the best understood microbiome now. And so there's a lot of very cool work that's being done. So we have lots of microbes associated with us that keep us healthy. And one of the things I wanted to mention was that what we're doing in the study of this, we're doing what other biology groups have done when they're faced with something really complex. And that is when you're faced with this kind of complexity, what you do is you go to model systems like Drosophila, like the fruit fly, or you go to C. elegans, the worm, or whatever. So what I'm showing here are some of the models of symbiosis that have been developed in the last few years. And these guys are models that are, oftentimes more simple than humans. The mouse isn't necessarily, and the zebrafish isn't necessarily, but they're good models of vertebrates because they're vertebrates. But the others up there, I'm going to talk about the fact that oftentimes they're simple. But the idea is what is conserved over evolutionary time is likely to be important. And since all animal body plans arose in the ocean, and the ocean is now and it was thought to be over evolutionary history around 1 million bacterial cells per mil of seawater, they grew up in the background of a huge number of bacteria and had to figure out how to handle that sort of situation. So I wanted to mention here that the use of model systems has been incredibly powerful. I'm given the example of Nobel Prizes in developmental biology. And there have been 15 awarded in developmental biology, physiology, and medicine since 1901. I've got on the left there how many were awarded for model system research. And this is the number awarded for human research. And so you can see that the human research is so difficult. That isn't to say that the value of this doesn't, and part of the point of having these model systems is to reflect upon how humans work. And in my work, I'll show you, I use a little model, but I'm always saying to the human biologist, look there. You know, we'll find something and we'll say, you know, look there. And we've several times we've been able to give them an idea of what might be happening. So what are the emerging patterns that we've seen over the last 15 to 20 years? And so what we have seen in looking at animals and their microbes is that there's a complexity to the species that has a pattern. And in the invertebrates, and that's most of the, and I'll show you a graph of this, most of the things out there are invertebrates. And those without backbones, that is, oftentimes, you know, lots of things in the ocean, all those clams, all those crabs, all those things, invertebrates. And they have low complexity of their microbiome for the most part. And there are some exceptions. Termites and cockroaches are big exceptions to that. But termites and cockroaches still seem to maintain the same microbiome individual. In the vertebrates, the complexity is very high. It's a shared derived character of all vertebrates to carry a microbiome, a very complex microbiome. And so the locations, the relationships with cells, interestingly, invertebrates, which are incredibly, they make up, I'm going to show this in a minute, but they make up 95% of the diversity on Earth. They often have both intra and extracellular bacteria. And any given species usually has intracellular or extracellular, but sometimes they will have both. The vertebrates, in other words, vertebrates are fish, amphibians, birds, and mammals, and reptiles. Those are the vertebrates. It's a shared derived character to be really complex. And they never accept pathogens. They never, ever, ever have intracellular symbionts, except the mitochondrion. And nobody really has an idea of why that is the case. What is the sanction on the carriage of intracellular symbionts? There's one strange example in a newt or something in its egg, but it's by far and away very, very unusual. Now, the correlation with this has to do, one of the correlations with this is in whether or not they have an adaptive immune system. So all vertebrates, a shared derived character of all vertebrates is the carriage of an adaptive immune system, which is, there's a gene called RAG that was thought to have dropped in to the vertebrate line at the agnath and naphistome transition. In other words, from the vertebrates that didn't have any jaws, the vertebrates that did have jaws. It's thought that there was a gene that dropped in called RAG1 that was enabling selection on that gene, enabled something called VDJ recombination, which is what allows your adaptive immune system to do that powerful memory. In other words, when you get a pathogen, and that pathogen, or you get a vaccine, you mount an immune system response to that allows you the next time you're exposed to it to remember, oh my God, that's that thing that really that was really harmful to me, and you mount a response. And so it has been thought for a very long time that that the invertebrates don't have, well, they don't have an adaptive immune system of the kind that the vertebrates have, but whether or not they have memory is still a question mark. there are lots of situations in the invertebrates that experiments have suggested that there is some kind of memory. And how that works most of the time is not well known. So, okay, the other thing is that about that was that the people who do biomedical research often don't know, they're trained in such a way that they don't know about the biological world in the sense that they don't know about invertebrates. They know about vertebrates and usually they just know about mammals, right? And so they had the mistaken notion that the immune system of invertebrates was very weak and that they couldn't do what they needed to do. And you know, it's really funny because there was a very famous immunologist who many of you will know, Charles Janeway, who wrote an article for Cell. And he said, well, invertebrates don't have an adaptive immune system because they're what is called R-selected. They live fast and die young. And so they don't need to. They don't need to do this. Well, in fact, yeah, obviously, you know, very, very famous guy, huge contributions, but he didn't know anything about invert zoo. Because there are clams that live in the mud in the Antarctic and lots of other places that live for 250 years, you know, and they are, you know, are just fine. And actually they have very few, if any, symbionts. So lots of, I guess my point is lots of misconceptions. So just to show you that the adaptive immune system is not correlated with biological success necessarily. So you have the adaptive immune system is what's present in the vertebrates, and they make up only 5% for the animal kingdom. All the rest of these things have what is thought only to be the innate immune system. So obviously, though, what they're doing is they're interacting with the microbial world in a different way, I think, than vertebrates do. So it's old news that adaptive immunity is an ancient immune strategy. So another misconception that people often have because they don't know about the geological timescale and the evolution of animals is that vertebrates are new things and that mammals are new things. They're not. Vertebrates arose with all the rest of the other phyla somewhere sort of Ordovician in the back 400 to 550 million years ago. And so they all arose about the same time and diversified. And so the fishes, which are the first evolved vertebrates, arose not too long after all the other invertebrates diverged. So to have an adaptive immune system is an ancient character. Is the thing. So here's the NIH website that I went to, prevailing definitions of immune systems. So what they say is that the innate immune system is the body's first line of defense against germs entering the body. Everything's about germs. They're not thinking about the fact that germs come into a pre-existing conversation that you have with your normal microbiome. And oftentimes I'll be talking to physicians and I'll say to them, how many pathogenic, how many times have you gotten a pathogenic infection? And they'll, you know, in your life. You know, they'll say, oh, well, 20, 30, something like that. Every day of your life, you're interacting with, hundreds of thousands of bacteria that you have to manage. So, you know, the pathogens can't be the only thing, right? So it's defense against germs entering the body. It responds in the same way to all germs and foreign substances, which is why it is sometimes referred to as the non-specific immune system. So keep that non-specific in your head because I'm going to be talking about this in a minute. The adaptive immune system or specific immune system is composed of specialized systemic cells and processes that eliminate pathogens or prevent their growth. And so these are things, that page was edited just like a few months ago. And so this continues to be something that's prevailing. And it's very interesting because the person who runs meta-organisms back at the NIH, or who did run meta-organisms back at the NIH, Jasmine Belcade, was of this view until several years ago. And she realized you can't think like that anymore. And now she says that the principal driving selection on the adaptive immune system of vertebrates is the carriage of a complex consortium and the management of that consortium. And so she's now actually moved to be the head of the Pasteur, so she's not at NIH anymore. So let's continue to look at these patterns of occurrence. So I'm showing on the left-hand side here a bunch of these invertebrate models. And so what's interesting about the fruit fly is that it has, instead of having, and this has nothing to do with size, I should tell you how many microbes they have, nothing, has nothing to do with size, because that guy down on the bottom is like this big, he's a big thing. And microbes are tiny, tiny. But the one up at the top of the fruit fly, depends on who you talk to. They have somewhere between 2 and 5 bacteria that they maintain in their gut, as you know, reproducibly individual to individual. You know, it's something that is fairly stable in their gut. Hydra, it's something between 12 and 20, depending upon the species. Honeybee is really interesting, 5. So a worker honeybee, if you talk to Nancy Moran, who's the major player in this field, ask her about honeybee, 5, reproducibly individual to individual honeybee, which is amazing. And that doesn't, and if you go to another hive, it's the same 5. You go to another hive, it's the same 5. If you look at a bumblebee, it's the same 5 except one different species. One of the species is different. So it's a really fun thing. They maintain 5. And the bed tube worm has 1 or 2 or 3, and those are intracellular. The fruit fly, hydra, and honeybee are extracellular. The bed tube worm is intracellular, and then Jack Gilbert out of UC San Diego did the European cuttlefish. And he found reproducibly individual to individual when he starved the cuttlefish. He found 2 phylotypes associated with them. They're like this big. 2 phylotypes associated with them. One was a Vibrio associated with the esophagus, and the other was a Rickettsia intracellular in gill tissue. So the Vibrio is extracellular. So it's pretty amazing. They have these reproducibly individual to individual, rejecting all other microbes out there, right? So what about the vertebrates? The vertebrates, in contrast, have a complex consortium of microbes. And that the carriage of that complex consortium is a shared derived character of all vertebrates. And so it's a strategy that the vertebrate body plan has. So let's connect these two things, the microbiome and the immune system. So what are we seeing and how is this changing? Well, before the advent of next-gen sequencing and before knowing about the microbiome, this was the prevailing idea. And this, I would say this, you know what, I couldn't put dates on this. And the reason I couldn't put dates on this is because this stays around till today. And so I actually went to a meeting at the Salk about 2 months ago. And I got up and talked about the microbiome and things like this. And after I sat down, there were, 50 more talks or something. And the microbiome was never mentioned once. They only talked about the pathogen that was associated with the system and how the immune system was dealing with the pathogen. So this continues today, but since next-gen sequencing, we become more and more aware of the microbiome. It's changing. The idea, the biomedical community called these bacteria commensal if they were in your gut. So people knew that you had microbes in your gut, but they called them commensal. And by the strict definition applied to the word commensal, what that means is that they have no effect on the host. It's a positive, so it's a fitness definition. So a mutualism is positive for both partners. A pathogenesis is positive for one partner and negative for the fitness of the other partner. And a commensal is that one partner benefits and the other is unharmed. So they thought these bacteria just were passing through and maybe digesting some of the food, but they were commensal. They didn't impact the fitness of the host. And so the non-self-recognition system or adaptive immune system was all about dealing with pathogens. For a very brief time, after we began to do next-gen sequencing, and Rob, you can correct me if I'm wrong on I would go to meetings and I would hear people say that we thought that there may be a shared microbiome that was the human microbiome. And it went on like this, actually, like I say, for a very brief time, because we became aware very quickly that this was unlikely to be true. There is a human fingerprint on the microbiome, but we, like you and I, don't have the same microbiome. It turns out that in vertebrates, the microbiome is more like a fingerprint. And they're individual taxon specific and highly complex and dynamic. And so instead of doing this to microbes, what the adaptive immune system allows us to do is be extremely permissive. The adaptive immune system, lots of the cells change over, 2-3 days. It's very fast, right? And so they're always surveilling. The adaptive immune system is extremely active, allowing, saying, can I let this one in? Can I let that one in? Can I, in other words, it's a very, very permissive. Rather than being restrictive, it's very permissive. And so the adaptive immune system's major purpose is to control the guilds in the microbiome. So it's a management system rather than saying no. Now, what about the invertebrates? What are the patterns we see? Well, very, 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 very different. It's extremely common, as I mentioned earlier. They are simple, stable, and invariant individual. It's just the way that the invertebrate systems seem to work between individuals and over a lifetime. And so it's really amazing. I'm showing a group down there at the left. Those guys were models that were developed of symbiosis. And symbiosis, I can tell you, guys, I was in the biggest backwater field going when I was a junior professor. There were very few people who studied symbiosis. There were oral people, or people like me who studied luminous bacterial symbioses. There were people who studied aphids and other insects. There was insects and microbiome. Insect symbiosis was very big because it allows them to live in environments and on food sources that are low in nutrients and hydrophilic. So these were some of the early symbiosis systems. And then once next-gen sequencing came around, we added a bunch of invertebrates, many of which I've already mentioned. And what these guys, what they have to do is in light of having these simple, stable, invariant situations, they have to repel all these guys that are out in the environment. As I mentioned, 10 to the 6 bacteria per mil, we can see them that these guys are interacting with every day, all the time. And if they're marine. So the invertebrate immune system, the innate system, behaves very differently. I want to give one example here, and that's Thomas Bosch's work, which I find really breathtaking. So Thomas is a developmental biologist. He's actually at University of Kiel, Germany, although he got his degree at UC Irvine here, his PhD. And he studies this little tiny, very, I don't want to say primitive, what's the word? It's a basal group of the animals. And it's like an anemone. And it has on its surface, he had a graduate student who came in and said, I'd like to look at the microbiome of Hydra. And he humored this student. In his lab, for 30 years, Thomas had 3 species of Hydra. And this student began to look at these 3 species of Hydra and their microbiome. And all 3 species, they had a different cohort. They were different genetic backgrounds. But all the Hydra viridis had the same 12 on their column. All the Hydra oligactus had the same, I don't remember how many there were, and viridis had the same number. And so it was an amazing thing. You would find them reproducibly individual. But what I really loved about what he did was he went back out into nature 30 years later and collected these guys again. And darned if they didn't have the exact same microbiome that they had in the lab. So that was tremendous fidelity over time in this invertebrate system. So it's a new day. We have these changing views of the immune system. And this is the Banksy that I love to show because it's like the elephant in the room. Nobody's noticing it, but what we're doing and what we must do is recalibrate our view of the immune system in animals. So I want to give one last example because I love it. It's what my lab works on. And this is the Hawaiian bobtail squid. And what this little guy does is the Hawaiian bobtail squid buries in the sand every day. And so in the morning when the sun comes up, it buries in the sand. And we have them in the lab. If you guys ever visit Caltech, I can show you around. And this guy's burying. And as soon as the sun goes down, Euprymna, this bobtail squid, Euprymna strophes, Euprymna comes out to forage in the water column. And it has associated with it luminous bacteria that it shines out of the bottom surface of its body. And that he manages the light production to match downwelling moonlight and starlight. So he's not seen by a visual predator looking up from below. So it's like a Klingon cloaking device or something. So anyway, it's an anti-predatory strategy. But what's great about these guys is we can have them in the lab non-symbiotic and they're perfectly healthy because it's not a nutrient they're given, it's light production. And what you see is that if this is an adult and they're only that big, they're only a couple of centimeters long. And if you do a ventral dissection of these guys, you see this bilobed organ in the center of the mantle cavity here. And this bilobed organ is actually a riff on the eye. In other words, because it handles light, lots of the biochemistry development and so on is very similar to the eye. And these are bioluminescent symbionts that have been plated out on agar. And the picture is taken in the dark, and they're Vibrio fischeri. So they're related to Vibrio cholera. So why do we like this symbiosis so much? Well, symbiosis is binary. And what kind of resolution is that? I mean, whenever that bacterium does something to the animal, you know that it's that bacteria. And you can make mutants in the bacteria in very special features like light production. And when light production is abrogated, the animal gets rid of the symbiote. It says, I'm sorry, you're not doing your job, you're gone. So it'll allow it to come in for 24 hours and then it recognizes it's not doing its job and it gets rid of it. And interestingly, the light production, not the presence of the bacteria, is the biggest driver of change in gene expression on symbiosis. So it's a very interesting thing, but we like it because it allows us to listen in with high resolution on the dialogue. How many of you guys have seen My Dinner with Andre? It's so great. I love this movie. Not a lot of action, I have to say, but what happens in this movie is you go in and these two guys start to talk and you think the guy on the left is a little bit crazy and the guy on the right is, very sane. By the end of the dinner, you sort of switch your idea about what's going on. And it's a very, and you understand the motivations of it, and you understand what's gone on in that conversation. And I liken the gut to the Kumbh Mela, beautiful Kumbh Mela celebration in India, where you've got 110 million Hindus, milling around, and try to figure out what the relationships and what they're doing is a very, very, very tough one. So I'm sort of simple-minded, so I really like looking at Euprymna. Also, as I mentioned, things are highly conserved over evolutionary time. And so there's a portion of our system that is mucociliary, and there's a portion that's microbial, which is very similar to, in their relationships to the bacteria, which is very similar to what goes on in the intestine and brachial epithelium. So we've been able to tell the biomedical community where to look on occasion. So for instance, we found that there were some molecules of the bacteria that drive development of the squid. And they are the exact same molecules that drive development of the gut-associated lymphoid tissue or some of the gut immune system. So that was fun. We study all aspects of symbiosis establishment. Now, our symbiotic systems are acquired into each generation when you pass through the birth canal and you get it from your mother and your family and so on and so forth. So you acquired it each generation, and that's what this animal does. So that's one of the big things that we look at. We look at development and how in the world do you keep them stable? The immune system doesn't get rid of them or they don't overgrow the host. And then figuring out how in the world, what are the differences with pathogenesis? And the immune system is important in all of these steps. And I'm going to give you one example here. And this is, I just got a minute or two. And this is them recruiting. So in the back, when they have to recruit their symbiont, this is a baby. The babies are like, 2 millimeters. And the baby hatches out, and within it faces a background of 1 million bacterial cells, for which Vibrio fischeri is about only 500 of them. So very, very low concentration. And within 12 hours, they have a pure culture inside their library. So somehow they're able to pick these guys out, or the bacteria pick them out. I think it's believed. So but what we've learned, we're studying this for a few decades, is that there's a reciprocal biochemical and biomechanical and biochemical partner dialogue. So it's like spy versus spy. Are you the right guy? Yeah, I'm the right guy. Are you sure you're the right guy? Yeah, I'm sure. Yeah, are you sure you're sure? And so it all the way along, they're sieving and getting just that particular microbe. And so, so you get over a dozen exchanges, and it's only over 120 microns, and the bacteria are 2 microns, so it's 60 body lengths of the bacteria, and they get a full organ by 12 hours. Now, this behavior, this way cool behavior, in my opinion, has happened with fidelity for 40 million years, or about 160 million generations with fidelity. I mean, that's amazing to me. They do this again and again and again. And so let's think about this. The NIH website says the innate immune system is non-specific. I don't think so. So with that, I'll just summarize and say, most fields of biology have been able to build on decades or hundreds of years of foundational work. So I give a quote here about the nervous system from a piece on the history of studying the nervous system. The understanding of human behavior as a result of brain-based functioning can be clearly traced back to at least the ancient Greek philosophers, and most likely even prior to that point, to the end of the Stone Age and ancient Egyptian times. So the studying neurobiology has captivated biologists for many, forever. And we just become more and more refined, and we're getting to the point where we understand the wiring and how things work. But we've been doing this for a very long time. In contrast, the discovery of the microbiome nearly instantaneously has changed our view of the biosphere. I mean, it's a very, very sharp change. 7,000 species to millions and millions. And the microbiome, the fact that the microbiome influences the form and function of all animal organ systems and plant organ systems and all corners of the natural world. And this is a huge, huge conceptual shift. And why is this conceptual shift so important at this time? Well, we're in a climate crisis, as we all know. And in my opinion, pushing forward this revolution is critical because we cannot be effective and efficient with facing these challenges if we have an inaccurate conceptual framework associated with the structure and function of the biological world. So with that, this was the 30th anniversary a couple of years ago in the study of the squid Vibrio system down at Scripps Oceanography, where we started. And these were some of the students and postdocs and faculty who could come back and be with us at this time, and the funding agencies that have supported us. So with that, I will stop and take questions.
We have lots of time, so we're just going to start here.
And this is a question from an absolute non-specialist.
Okay,
And this was prompted by your reference to Galileo's use of the telescope. As we know, in that case, there were people disagreeing.
oh, yeah,
And from your interview in San Diego, I understood that some people also disagree. So I was wondering whether there are discussions by people who completely reject these views, or whether all those who basically were aware of what you have presented accept these views.
they don't reject them, they ignore them, in my opinion. And what it is that, unlike a lot of fields, biology has been very siloed since the 1950s. And that's reflected in the structure of departments, in the structure of funding agencies, and so on. So what you see that people will, as undergraduates and graduate students, get degrees in cell biology and not know anything about, they may have a PhD and work in something on organisms like Dictyostelium, which is another, Chlamydomonas, which is another, but they really don't know as well. In other words, there's a lot of siloing in biology, tremendous amount, that is hard to overcome. And I think, I personally think that the remedy is education, and is, when I was at Caltech, I had this fabulous opportunity as a Moore Fellow to teach a biology course. So I've taught intro bio maybe 18 times. And I counted them up, it is 18 times. And 2 of those were at Caltech. And I did those 2 with a microbiologist named Diane Newman. And she was the person who did the microbiology. So every class, I would stand up, she would stand up and say, okay, we're going to do transcriptional regulation or behavior or ecology. And she'd start with the microbial world. How has it historically and how does it now work in the microbiological world? And then I would get up afterwards and I say, well, okay, because I'm an animal biologist, I'd say, okay, now that you've got a nucleus in the cell, and now that you've got animals and you're multicellular, what is the nuance that you put onto that that is that onto the microbial? So there are a couple of examples that are really stark. I mean, if you ask most biologists who are not microbiologists, you can ask them the question, what is the terminal electron receptor? Without exception, I can tell you they will say oxygen. Oxygen is the terminal electron receptor. Well, if you understood the microbial world, you would understand what is possible with the physical environment that is there to begin with, the Earth. I mean, on Earth, what's possible? There are, I think, 10 or 12, I always forget how many, terminal electron acceptors and donors and acceptors that are used by the microbial world. And it's just that when eukaryotes evolved, and that was, oxygen is biggest bang for your buck. There's no question about that. And the mitochondrion was taken into the host cell, and you got the eukaryotic cell. That's what allowed the radiation of eukaryotes. And so it's just one single terminal electron acceptor, but you don't really understand the concept of terminal electron acceptors if it's possible with the Earth. Another good example, in my opinion, is behavior. Bacteria have on their front end, they have these chemoreceptors, and those chemoreceptors tell them what's out there. And so what they have to do is say there's food out there and they want to chemotax toward it. They want to move toward it. They sense it, and then they have an integration across the cell that tells them whether or not they should move to that or whether or not it's poison in their, what in their life history and in their evolution, what is that good or bad? So they have these receptors, they have an integration, phosphorylation cascade usually, and then they have an effector, which on the other end is a flagellum. And so they move for that. So Diane would get up and she'd talk about all this biochemistry that was associated with that. And then I would get up and I'd say, okay, now that you're an animal, what do you do? Well, there are three kinds of neurons. There are sensory neurons, interneurons, and motor neurons. And then I would go into how action potential works and all this other sort of stuff. But, if we could teach, so another little exercise I've done is to go to the textbooks of chemists and physicists. And I'm an old girl, and I've gone back to the ones that I had when I was in college. The same big table of contents is present in the chemistry and physics textbook. It's just this, electromagnetism, whatever. The same big table of contents is present. You do the same thing for a biology book, and there's nothing even close. Biology textbooks are bricolage, and I personally believe, and Diane Newman and I, strove to do this. I personally believe that we can now unify biology in ways that chemistry and physics has done, should we be willing to integrate macro and microbiology and build, in that way, students in an introductory course get the history of the biosphere, evolution, way things work at the molecular level, and then all the way up to ecology. And so you ought to be able to teach that over a year, and then people can wing off the way they do in chemistry and physics into cell biology and whatnot, but they have a framework, they have a foundation in the field. And I personally think that that's where we need to go.
I think I have a second question over here. So I wanted to ask a little bit about the opening framing and like how far you want to take that with respect to the microbiome. So in invoking the Copernican revolution, one of the things that is highlighted, and you mentioned it, is that it somehow changes the way we think about ourselves. And for the microbiome, there's been a little bit of literature over the past 7 or 8 years where pretty radical claims about the way in which the microbiome is supposed to reconfigure our understanding of ourselves has sort of come into question. People have said this is a bit overreach. So there was a paper that actually argued for the reconfiguration of the humanities in light of the microbiome. And it was rightly like, maybe not. So I guess I'm wondering, in thinking about the revolutionary nature of the microbiome and the kinds of things that clearly do have wide-reaching effects on how we think about the living systems around us, how do you think about maybe when it's too far and when it's too much to say that the microbiome affects this too? Or
Yeah, I personally think that's too far. Yeah, I believe that the microbiome is like I said, like the 11th human organ system or the 11th cat organ system or whatever. In other words, there are things that associate, from that viewpoint. So the microbiome is not only, relevant to thinking about animals and plants, but the soil. Are we going to think about the soil completely differently? We know that I feel like we should integrate this information into, I have trouble going there, I guess. It's just, it doesn't resonate with me, but it's, maybe because I'm a biologist and, I look at it very mechanistically.
So I had a question. I was intrigued by this list of species that you went through that had certain bacteria that they preferred, or rather settled.
Always occur.
And you say that that this particular kind of bumblebee likes these kind of bacteria, 1, 2, 3, 4, 5, and that's like their favorite thing. And if you go to another bee, it's slightly different, but there's stability there. So you've identified a pattern in nature, but coming from a physics perspective, I would say, can you give a reason why the bumblebee likes these particular bacteria. In other words, not only to identify the pattern, but to give a reason for the pattern.
Yeah, so what Nancy Moran has been doing with the system has been taking one away and see what happens. You can culture all of these, you can, you can make, I think she's got genetics in most, if not all of them. And so she can manipulate them genetically and find out what's essential and so on. I look at it as, that's a very interesting question. And I just look at it, like I said, mechanistically, how does it work? It obviously arose through evolution. And that evolution was likely something that was a complex conversation between that consortium and those ancestral honeybees. One of the things that can happen is you'll look at the microbe and so let's say you have a group of animals that all have similar microbiomes. And you look at their phylogenetic tree, and the phylogenetic tree often will be congruent with the phylogenetic tree of the microbes. And so obviously this has arisen together. It's very commonly functional. In other words, what they're doing for each other, I guess. Rob, do you have any thoughts on that?
Well, I think that I think that equations together mechanism for a lot of these things already is kind of unreasonable given the time scale, right? Like if you look at, say, the discovery of Saturn's rings to explanations for how they formed or how old they are, that's hundreds of years. So given that with these species and these patterns, we're talking about maybe a 10-year horizon when the patterns were discovered. It's probably going to take a while to develop mechanisms.
Mm-hmm. In other words, why that far?
Yeah, well, the uncharitable view from the mechanistic physicist would be to say, well, there's physics and everything else is butterfly collecting. So I'd like to see from a biological point of view, can you go beyond just classifying or seeing patterns to finding reasons for it?
Yeah, well, in the system I work on, obviously the pattern is, the presence of a light-producing bacteria. And the reason that it's there is anti-predation.
That's one example. Yeah, let's go to another question.
Just one comment and a question. The comment is I've been taking pictures of every slide because my wife's a retina surgeon. There will be a real question, but my wife's a retina surgeon and a neuroscientist who works on mular cells in the eye, but she's discovered microbiomes the last 5 years. And like every dinner, we talk about the gut and other bacteria. So thank you very much. But she flies basically 40 weeks around the world to give talks about why this matters for her little niche in medicine.
Mm-hmm.
So I think people are picking up on it.
Yeah, it's a hard thing.
It's a hard sell, but they are picking up on it. My question is about your plan for unification at the end. And I could see the attraction of it, particularly pedagogically and also almost humanistically, as you yourself suggest. But one theme that has emerged from the conference is that getting people in different fields and niches talking to each other, even within big disciplines, requires an enormous amount of mediation and translation. And because the way you want to unify actually doesn't provide really a translation model or even a really deep or theory of how these things hang together.
Mm-hmm.
And I can imagine that there won't just be people protecting turf resistance, which is what you're encountering on ignorance, but also a sense that actually there are too big of an opportunity cost to do all this generalist education in the first year. Whereas to become this hyper specialist or even well-educated cell biologist, they need just too much other knowledge. And so while I agree with you that it would be great to integrate, I haven't really heard you address what I think are really quite reasonable potential objections to this kind of.
Yeah, I think that what you're aiming for in this unification would be talking to biologists about things like thermodynamics. Why do biological systems seemingly defy the laws of thermodynamics? Well, they don't, right? There's a couple of reactions. But the fact of the matter is I think rather than what intro bio at this point is a lot of memorization. And so we taught this course at Caltech. It was not to the biology majors, which got cell and molecular biology at Caltech, as you might imagine. Instead, this was the astronomers, the chemists, the physicists, the mathematicians, computer scientists that we were teaching. And so at the end of the day, we started with the history of the Earth. They got a very good idea of how life evolved, the great oxygenation events, and then, down into evolution, which is the glue that joins all biology, getting a good, cell biologists often don't get any evolutionary biology. It's like ridiculous. That's the glue that joins you all. And it's what you want is you want the biologist to be enabled and to really, understand the basic principles of biological systems, the way physicists and chemists understand the basic systems. And I think it's doable. It's not easy.
But I didn't hear principles. I heard, so what I'm trying to get at is, this is really, nothing I want to say is to diminish your project, but I haven't quite heard yet what the principles are, other than really a lot of, not like a collection of facts, but a lot of interesting systems that you're studying. But what the principles are that actually tie those different systems together.
Energetics.
It's not entirely evident yet. It's Darwinism, it's ecology, it's symbiotic, but you haven't conveyed to me, perhaps because I'm ignorant.
Well, no, it's another talk.
Okay.
Actually, yeah, I've been asked to give this talk. I gave it at UT Austin, and it's like, it's things like energetics, and there are things that you want all biologists to know and understand how things work at various levels. And you're right. The stuff that I talked about today is such a slice of that. So the reason why integrating microbes into our understanding is that you start with the fact that you're looking at an organism whose behavior and ecology are their biochemistry and molecular biology. So you have this starting point in which the hierarchy of life is collapsed. And so you look at how it's done in that situation, and then you become more complex. But it's not an easy thing. And, I think it's just something that I feel would be an exciting thing to think about.
I take the class.
Well, we'll see what happens. Actually, this is really funny. After I taught this class, the president of Caltech always has a party for all the faculty at the end of the year. So I go to this party, and one of the members of the faculty who taught intro bio to the bio majors walked up to me and said, "So do you think your chemists and physicists and mathematicians know any biology?" And I said, "I'd put them up against yours anytime." Anytime.
I think Jose, you had a question?
Yes, so apparently these discoveries of all the variety in all those different forms of life, they present a very radical different picture than the one that we had before. So my question is, thinking about the climate crisis in which we are, is this picture more robust against the consequences of the climate change? Or is it more weaker, or it's not going to affect, or it's more difficult to predict? So what is your opinion?
Well, I think that that there is a lot of evidence that the oceans and the soil and whatnot, those really important ecologies, regions, are run at their foundation by the microbial world. And so it's really important. I was just up at Stanford talking to a guy who works on water relations of trees and forests. And I said to him, are you studying the mycorrhizae? And he said, no, that's not important. Mycorrhizae are what control water relations in the soil. And I was like, well, okay. It's an interesting thing. And the old saying, Science moves forward one funeral at a time.
Yeah.
You know, I am known as somebody who is trying. But
Okay, mine?
Yes, to your comment, so I'm part of and to your comment, so I'm part of a biology integration institute where we have microbiologists, we have systems biologists, we have computational biologists, but then you have geologists, we have computer scientists, because you simply can't understand the whole biome system. But without work like this, you can't understand the small, you have to start at the basics and build out. And then you can't, challenges, anthropomorphic change is unprecedented on the time scale. You can't understand how the system changes unless you understand how at least some of the variables work. So my question to you, Margaret, is what's the next arrow for the next thing that we don't have?
yeah, a good
Because
question.
it's clear in these models that we miss something, because
Yeah.
you're trying to model input and output,
It's very, yeah,
and there's something missing still.
yeah, well, and it's really short time
Yeah,
frame, and so things are gonna keep morphing. You're
Yeah.
right, and I'm not sure what's gonna be next. But you know, one of the things you mentioned earlier in your question, and one of the reasons why I think we have to do this is that many of the problems of humankind have biology at their base, if not the majority of them. But chemistry, physics, math, and so on at their solutions. And those guys, at least when I was a kid, didn't have to take biology. And so I collaborate with physicists and biomechanics people who have very little biology, and speaking the language and communicating with them is very difficult. And so I think that getting biology courses in which, I mean, I had to take chemistry and physics with the chem and physics majors, you know, and I think that it's important that we do that as well.
but you don't know what the next big arrow.
I,
Because the thing with the sequencing is it's getting cheaper and cheaper and better, but it's the same information at higher and higher resolution, bigger and bigger quantities, some of it.
yeah, there are big messages that have been coming out.
Yeah.
Like one of the big messages is we were talking about the immune system, that the things that run the immune system, like what has been called antimicrobial peptides, and all those things that we've ascribed to being antimicrobial we now know are things that actually run the system. And so there's some push to change the lexicon, you know, and that's, you know, and you're right. I mean, people are beginning to look at mechanism. And so one of the things, because it's so complicated, is using these models. And as the models, you know, as we get more and more sophisticated with those, it's actually very interesting what you can learn about looking at these models. You know, Thomas Bosch, that guy who did that thing, he showed that Hydra gets cancer. Okay, Hydra gets cancer, they get a big tumor. And it turns out that he made them with no microbes on them. And if you he was able to find which bug was in the cancer and that bug caused the cancer. But then he showed if you have no microbes and he was doing Koch's postulates, he took all the microbes away from the surface with antibiotics and then gave that particular cancer causing microbe, and it didn't cause cancer. It requires one of the members of the microbiota to be present. And so, you know, that's just a very simple sort of thing. But all these things like Nancy Moran's work with the honeybees, if you've got a consortium of 5 bacteria, you can begin to say, how are things managed? You know, what do you do when you have more than one? When you have 2, when you have 3, when you have 4, when you have 5? How do you do that? It's so complicated. You know, it's like the nervous system was, you know, so 1990s were the decade of the brain. I remember being at USC, 1990s were the decade of the brain. They had a lot of trouble because we weren't quite there with the computer science and stuff like that, and AI. and whatnot, and now great strides are being made. And so maybe, maybe it's gonna be something like that.
Okay, I think we're out of time, so let's take the rest of the questions in the coffee break. We can thank Margaret again.